And thank you for attending. I'm Val Valev. I'm a professor in bioengineering. And we've been waiting for this lecture to happen for many years. And it was in preparation and always postponed because of obvious events since 2020, since the beginning of 2020. And there were some other events that postponed until now, but finally it's happening. So welcome everybody to the inaugural lecture on Jerry and Jane Schultz's honored lectureship. And before we go into the program, this lecture is recorded and there is also a webinar component. So people online, you can write down your questions and in the end we'll read them out loud and the speaker will answer them. So we're very pleased to have Eric Betzig. He's a Nobel laureate in chemistry in 2014. But before we get to the lecture, I would like to introduce the Dean of Engineering of Barnes College of Engineering, Chris Lynch, to give a few introductory remarks.
Thank you so much, Val. And I'll tell you, we couldn't be more honored than to be launching this inaugural event of the Jerome Schultz lecture series. Jerry Schultz was the founding chair of our bioengineering program. And to come back and help us out like this and help promote the program is just really important at this moment in time. Since I've been here in 2018, we've been under a period of very rapid growth. We've gone from 3,400 students to 5,000 students this fall. We've expanded a number of programs, including bioengineering. And one of the critical strategic areas that we're looking at is the intersection of engineering with medicine and life sciences. There are just so many discoveries in the life sciences that with the engineering technology coming together, and this applies as well to medicine and agriculture, it's transformational. Our bioengineering department is poised to really make a difference. In the 5 years I've been here, their student body has grown 33%. The number of applications to the program is way up. And we're really hiring a lot of faculty. We're looking to expand. This is an area that's made a huge difference. And Jerry, you started this. Really appreciate it. So I want to leave the time really for our guest of honor, the speaker. So let me turn it back over to Val to introduce the next person. Thank you.
Okay. Thank you, Chris. And as you said, Jerry was the founding chair, but unfortunately he's not the chair anymore. So we have another person who can actually talk about the present state of the department. So that's Xiaoping Hu, who has been chair since before the pandemic. So pretty much you coincide.
Okay, thank you. On behalf of the bioengineering department, I want to welcome everybody here. This is really a special event. We have been looking forward to it. Our department, as Dean mentioned, has been growing, not only in number of students, but also in the faculty. And we are close to 20 faculty now. When I joined, it was about 12. So there is a big growth, and in response not only to the student growth, but also research opportunities, particularly research opportunities from the NIH and also in collaborations with medicine. So I want to say that, this department would not be possible without Jerry, who has been instrumental, and also other senior faculty members and the leadership like Victor and all those people. So I want to thank them for the, for the sort of growth of our department. And of course, this is a special event for us because we are very grateful to Jerry and also, for setting up this lecture. I will not spend more time. I will just introduce Jerry, who has been an instrumental person to our department and whose generous support without which this lecture would not be possible. Jerry?
Thank you. So, speaking about the department started with 4 of us, actually, and don't remember exactly the date, but Val, Victor Bauman is here, and Dimitri, so the 4 horsemen, and Jayu started the department. And Yeah, it was my goal at the time to have a department of 20. We didn't quite reach it at that point, but I'm glad to see the department growing. But anyway, so I'm talking too much. But when Val told me that Dr. Betzig was going to be the inaugural speaker, of course, I immediately went to the web to see what about this gentleman. And I came across this picture, which was actually on the website of the Nobel Prize Foundation. But what got my attention, in addition to the work he's doing, is that he was born in Ann Arbor, Michigan. And it turns out that, how do I do this? That actually I was in Ann Arbor, Michigan when he was born. But this picture, and I don't know if it's true, but it was on the web, it was his senior picture at Pioneer High School. And it turns out that my two kids, Danny and Judy, twins, went to Ewing High School, which was the competing high school for Pioneer. I don't know which football team won at that date, but anyway, I'm sure the kids crossed paths. And that was me in that time, and when I was chairman of chemical engineering. And here's a brief summary of Dr. Betzig's career. Started in Pioneer High School, and no other introduction starts with high school, but, but started Pioneer High School in Ann Arbor, then went to Caltech. I asked him why he didn't go to University of Michigan, and he said that his other members of his family went to he was a rebel. They went to the University of Michigan. He went to Caltech, then on to Cornell, and then on to Bell Laboratories, and then was a stay-at-home dad. That's your quote. And actually, it was during this period of 89, roughly, to 96, that he really came and conceived and determined the basis for eventually the Nobel Prize. Then he went on to start a company for a little while, to a research group sponsored by the Howard Hughes Foundation, and then UC Berkeley. And with that brief introduction, we want to get on to the speech. So I don't know why that did that, but here we go. And so I welcome to the podium Dr. Betzig, and we want to learn more about your window to the cells. Did you want to have a picture at this point, or
We are supposed to have a picture.
we're supposed to have a picture of the four of us?
Set up for a picture at this point.
I didn't try the program.
But that was a program you sent me.
Yeah, but there was no fault. Okay. Yeah. Shall we put the microphone somewhere?
Oh, here's the part.
Yeah.
Look at,
All cameras.
yeah. Okay.
Okay.
Thank you.
Is it sound? Is that work? Let's see. Is it, how do you turn it on? I could hear it now. Yep, that sounds good. Everybody can hear? All right, good. Oh, okay, looks good. Oh, yeah, that wouldn't be a bad idea. Thank you very much. Good. All right, well, anyway, thank you everybody for coming, and thank you, Val, for dragging me down here. And I don't really give many talks anymore. I've kind of burnt out on it. And I have still young kids at home, and I like being in the lab, but because one of my closest friends and collaborators, Gokul Padahayalua, got his PhD in Val's group, I wanted to come and pay my respects, and it was good to hear about Jerry and his history, and I'm really very grateful that you've started this series and that I'm honored to be your inaugural speaker. So the microscope has been around for about 400 years, and for 300 of those years, it was the predominant tool by which we understood living things below the level of the naked eye. And early in the 17th century, Robert Hooke made a microscope. He looked at many things from fleas to flowers and wrote this book, Micrographia, which was, other than the Bible, the very first bestseller. Not the first scientific bestseller, the first bestseller of any kind. And people were captivated by what you could learn by looking through a microscope. Around the same time, or a little bit later, Antoni van Leeuwenhoek made a microscope that was different in design, which had even higher resolution, with which he was able to scrape the tartar off his teeth and see that there were little tiny rods, which were bacteria. So he's actually the father of microbiology. And his microscopes were amazing, but he kept secret the method of their manufacture. And so it was 150 years before anybody would have a microscope as good as von Leeuwenhoek's again. So microscopes did gradually improve and beat and surpass von Leeuwenhoek's microscope, in large measure to the efforts of these two guys in the end. Carl Zeiss was a manufacturer of microscopes in the 19th century, and he was frustrated with the irreproducibility of his manufacturing line. And so he got a physicist in the local university, Ernst Abbe, to try to look at the problem and see what was wrong. And Abbe came up with a theory for exactly how all the lens would have to be ground, in what shapes and what separations and what materials, in order to get optimal performance. And in fact, he found out that there was a limit to that performance. And that limit was basically half of the wavelength of light. That you wouldn't, no matter what you did, you wouldn't be able to get a microscope better than that. But his microscopes, his calculations were perfect. And when Zeiss followed those, with the help of Otto Schott, who was able to make better glasses, they did get to the diffraction limit back in the 19th century. And as a result, between 1880 and 1980, the microscope really didn't change much. I mean, certainly we had better light sources, we had incandescent lamps, we had filters, we had all sorts of other things, but the basic operation, the basic limits of the microscope were effectively unchanged for a century. So that changed, well, as a result of that, in the 20th century, there were different types of technologies that biologists turned to understand the workings of cells. So first was biochemistry, where you grind up your cells in a blender and see what the proteins and so forth are that it's made of and how they interact. The second was, of course, the revolution with molecular biology and the understanding of DNA and RNA and the whole paradigm about how proteins are created by these. And then also, starting in the 1930s with the development of the electron microscope by Ruska, and more recent things like with first X-ray crystallography and more recently cryo-electron tomography, to have a very, very detailed picture of things way beyond the optical diffraction limit. But again, not with the protein-specific contrast that optics provides. But I would argue, despite the power and how much more we know about living systems as a result of those three techniques, we still don't know enough. And part of that is because they're all extremely reductionist. You're taking a very complicated, holistic system and trying to break it down to its parts and reverse engineer to figure out that whole system works. But if I tried to do that for a BMW engine and I laid out all those parts, if I didn't have any prior knowledge of what a spark plug does or what a camshaft does, I wouldn't really be able to reverse engineer and figure that out. And the reality is even worse than that, because when you do things like biochemistry or structural biology, you don't even know that you have all the components there. And you don't even know if you have them in the stoichiometrically correct amounts. You might have 8 spark plugs instead of 6, and you don't know what's going on. And so it's been very informative, but we really need to see the cell in its entirety and holistically. So I had the dumb luck to enter graduate school in 1982 when that stagnation of the microscope for a century was about to be turned on its head. And it was due to several unrelated advances. First, the transistor in 1947, which led ultimately to the integrated circuit, which then led to the IBM PC. Computers that were cheap enough that you could put on a bench and have them automate your microscopes. The other thing that integrated circuits led to were semiconductor optical detectors of enormous sensitivity. So CCD cameras and now CMOS cameras have revolutionized our ability to see very weak signals coming from living systems. With particularly that imaging technology married to the computer and digital imaging, you then were able to now take movies and quantify things like the dynamics of whole cells like you see here. The next major advantage was around the same time when chemists started to get more and more clever about how to be able to introduce chemical compounds that were specific to attach to specific types of proteins in cells, so those particular parts of the cell, in this case the mitochondria in red, would light up. And so now all of a sudden we can have this protein-specific contrast, and since proteins drive the function of cells, we have a real new window into what's going on. And then the final piece of the puzzle was starting in the early 60s, the growth, development of the laser, which by the time of 1980 got to the point where you could have a tabletop laser in many different colors that you could afford to create very pure light to excite fluorescence, or even because it's coherent light, to change how you shape that light to make all sorts of novel types of microscopes that I will get into. And so pretty much since 1980, everything that's happened in my field has been nothing about a bunch of guys saying, okay, I can use these different ingredients and I can bake a different cake for a different microscope to do this, 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 or this. And this is what I've been doing for my career. So what are the different kinds of things you can do with those new tools? Well, the first thing that most people would think of, since when you think of microscope in your head, you think, oh, resolution. And you think about Abbe and you think about there's a limit. So first thing you'd like to do is, can you get past that limit? Well, what does that limit really mean, half the wavelength of light in biological terms? Well, if that green barrel represents a particular protein molecule, then the image of the protein molecule in your optical microscope at the diffraction limit looks like that. So you're off by about a factor of 100 of being able to see and at high density the individual proteins and how they interact to create cellular structure and drive cellular function. So there was a couple professors when I went to Cornell in 1982 who had a crazy idea about trying to make an optical microscope with the resolution of an electron microscope. And their idea was, Mike Isaacson was an electron microscopist who would do lithography to drill nanometric holes inside of silicon wafers. And Aaron Lewis was a Raman spectroscopist who thought, along with Aaron, well, if we took one of those silicon nitride windows that you have and coat it with an opaque metal, but it doesn't fill the hole, and the hole is smaller than the wavelength of light, then if we shine light on one side of that screen, then the light that comes out will be initially collimated to something much less than the wavelength of light, and we can use that as a little nano flashlight that we can scan point by point and get an optical image with the resolution of a electron microscope. Since optics can look at live cells, we could do live cell imaging. So that was the dream, and so that was my PhD project. And so eventually I built this sort of train wreck here as my first microscope, my near-field microscope for my PhD thesis. There was the IBM PCs running it. And eventually we sort of poked a little bit beyond the diffraction limit after 6 years of hard labor. And, you know, it really wasn't much, but it served its purpose, which was to get me a job at Bell Labs. So at that time, Bell Labs was still the premier research institution in the country. It was still to this day my favorite place I've ever worked. It just had a dynamism to it that I've never seen anywhere else. and brilliant people. And so after a lot of hard work, and I almost quit at one point because I wasn't making progress, but then I started to have a couple breakthroughs, again because of random people down the hall doing different things I didn't think would be useful, turned out to be very useful. And so at one point, we were able to do super resolution photolithography and photoresist. We were able to look at tissue sections beyond the diffraction limit. We were able to study how two different lipids change their organization under surface pressure. We were, at one point, had the world record for data storage density when we were writing bits this small versus the conventional diffraction limited bits that you see there. And eventually, although I was never able to get it to work on live cells, I was able to do fluorescence imaging of fixed cells and go from resolution like that to the actin cytoskeleton you see there. So those are all the victories. There were two other things that came out of this period, though. The first is there was a lot of buzz in the air at that time about single molecule detection. My co-lawyer, W.E. Warner, had around that time, by working near absolute zero, had seen the spectral signature of single molecules. And so the thought was, you know, can you see them optically at room temperature? And a lot of people were working on this, and the main problem is just the amount of background. The molecule itself produces a lot of signal, but at that scale, even a very weak emitting sample will create all sorts of background. Well, near field, you're constricting the light to a much smaller spot, so there's a lot less background when you excite. And so it became trivial. So this was an afternoon experiment that was a science paper. It was literally an afternoon. And so what we found is not only can you detect the molecules, but because of the polarization relative to the dipole moment of the molecule, you can actually determine the orientation of the molecules. You can actually determine their positions to a small fraction of even the near field size. You could localize them to about 10 nanometers, or, you know, about a factor of 100 past the diffraction limit. And then in another experiment I did with my best friend, Harold Hess at Bell Labs, he was a guy who was doing low temperature scanning tunneling microscopy. STM was a hot topic back at that time. And so one day we took his STM tip out of his cryostat and we put in a near field tip to be able to study the light emission inside of semiconductor multi-layers, like in a laser pointer, like a heterostructure like this. And what we found is that where the emission occurs in these layers was not uniform at all. Instead, it was coming from little discrete spots. And furthermore, the color that was coming out of each spot was differently. Now, the density of the spots was so high that even with that small near-field tip, there might be 100 glowing spots underneath the probe, and normally you wouldn't be able to resolve that. But because we were cold temperatures and the spectral lines were so sharp, we were able to resolve them by a combination of the high spatial resolution to have only 100, and then the high spectral resolution to see that each one of those guys glows in a different color. And so we could build up this multi-dimensional picture of X, Y, and color to be able to study those points of emission individually. Okay, so that was a really great experiment. And so, so that was all the victories. And then finally, I kept pushing to try to look at living cells with near field, and I could never get it to work. And ultimately, I realized I'm being an idiot for the last 12 years because this technique has some basic fundamental physical problems. And the first one is that, yes, the light that comes out of that hole is collimated, but man, it spreads like crazy, really, really fast. And if you do the math or you do the experiment, you can see that even by the time you're 25 nanometers, which is still, you know, like a 20th of the wavelength of light away, you're losing significant resolution. Well, I learned enough biology at that point to know that cells are not flat to 25 nanometers, so I wasn't really going to be able to get to my dream of being able to look at living cells with that. So I was really frustrated. In fact, if you looked at all those examples that I just showed you in the last two slides, they had one thing in common. The samples were exceedingly flat, and the thing you wanted to see was exceedingly close to the surface. And so that's all near field's good for. And so I got frustrated. I got frustrated by another thing. is that when I got into the field, everybody said, no, you can't beat Abbe's limit. I don't care about your stupid hole. No, it's never going to work, blah, 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 blah. And so you get into it and there's nothing but disbelief. And I said, I don't see any reason why this wouldn't work. And so eventually you start working on it and you start getting some successes and you say, hey, this is actually going to work. And you get really, really excited and you think, I'm going to take it all the way to here, okay? So you started going up the curve and you get better and better, as you could see when I was at Bell. And then eventually you kind of top out and you start to realize, oh no, there's actually some real limitations to this damn thing that I am not going to get past. And you realize the reality is much lower than what you would hope it would be. So, you know, the scales fall from your eyes and the dream is dead, right? But at the same time, because you've just published all of those papers and learned all of this the hard way, the field gets hot, and many, many people jump into it, right? I mean, just lots of bandwagon scientists out there, and they all plop on board, and they all say, everything's going to come out of this microscope. We're going to sequence DNA, we're going to do this, we're going to do that, da-da-da-da, and the hype just goes to infinity. And so, you know, I am not an academic, but, you know, I've just have always felt that you get to this point in the process when you realize the limitations. And I kind of felt like every good paper we published was just the justification for 100 pieces of crap to be published in its wake. And that everything I was doing was a net negative to society and a waste of the taxpayer's dollar. And so I was really in that state of mind when I said, I'm done with science, and I quit. So I quit Bell Labs right at my peak in terms of you know how well I was received within the building, and I didn't have a plan B. So that's when I was the house husband. Okay, so my wife had a job at Bell. We had a, let's see, at that time, a 1-year-old daughter, our first kid, and so I was just kind of figuring out what the hell I'm going to do next. And so I'm pushing the kid around in the stroller. And literally just from nowhere. You know, the thing is if you immerse yourself in anything and devote your soul to it, it's always in your head. I don't care what you're doing, it's in the background, in the subconscious, and the subconscious continues to work on the problem. And so I was pushing my down the stroller and I said, oh man, if we could combine this idea of localization from that single molecule experiment I did with this idea of discrimination by having an extra dimension, like we did in the semiconductor case, we could come up with a completely different way of doing super resolution. So the idea is you have a bunch of fluorescent molecules on a sample, and remember that fuzzy ball, how they look like a big fuzzy ball. Well, all the fuzzy balls run together in a normal diffraction limited image, and you can't do anything with that. Let's say those fuzzy balls, though, all glowed in a different color, for example. Well, then I could take a bunch of images with filters specifically for different colors, and then I start to build up this three-dimensional image where I have a fuzzy ball, but the fuzzy ball is now in 3D. And in 3D, it's separated from all the other fuzzy balls because I have that extra dimension. It hasn't been collapsed to 2D. Well, once they're isolated, then I can do the trick I did with the single molecule experiment and do a fit to the fuzzy ball to find its center. And that's where the molecule is. So then I find the molecules, and I then project those back down into the spatial coordinates, and I've got a super resolution image. Okay. So at the time, the only way I knew how to do that experiment would be by going back to Bell Labs and going to Harold and putting in his doer and trying to do this near absolute zero. And I thought, we could do it. It would be a really hard experiment. And God knows that, you know, you're not going to be looking at live cells at 2 degrees Kelvin. So this has got limited utility. So just thankfully, because I was stubborn, even though I was unemployed, I published that idea. And that was one of the 2 papers the Nobel Committee cited for giving me the prize. So I don't know what that is. That's the ad for this machine. So that's the little movie running in the corner. So in the end, one of the reasons that I would take these risks in order to like do something crazy like near field with those guys in '82, and why I would be crazy enough to quit Bell Labs, is because I always had a backup plan. Because my dad in Michigan had a machine tool company which in the 90s was growing like a weed. This was when the boomers were all buying SUVs like crazy and so forth, and so there was there's a lot of money to be had. So his company grew to be very large enough to be able to have a leech like me come on as VP of R&D. So I came on, and I also my dad made these very custom machines that would have to work 24 hours a day to produce millions of intake manifolds or brake calipers or what have you, and they were specifically designed to make one part. And CNCs, these programmable, flexible machines were around, but they just didn't have the robustness. And so I came up with a machine that would have the robustness yet the flexibility of CNCs, and extremely fast, extremely productive. I was extremely proud of everything I put into this machine over 6 years, and in the end I sold 2 of them. And so it was a complete disaster, well over $1 million of my dad's money down the toilet. And I did everything I knew how to do. It was just kind of like Muirfield. At some point you say, I've hit the wall bloody and I don't know how to go any farther, and so I quit. And then, so that was then the second time into the abyss, and I didn't know what I was going to do because now I'd blown up my safety net backup plan, so now what? So I realized, and I started talking with Harold now, because as Bell Labs finally imploded around 2000 or so, and Harold ended up going to work for a disk drive inspection company in San Diego, and we started talking again and saying, we don't really like academic science, but we really miss doing science with our own hands in our own lab. And so also everything I knew about science had kind of atrophied in the decade of working with my dad and being a house husband. And so I just started going back to my old freshman textbooks and started redoing homework problems and working my way up. And you realize the information is still there. It just is behind a wall and it has to come down. And then I started reading the scientific literature, and I knew 2 things going into this. I wanted to do science, and I didn't want to do microscopy because I had been there and done that. And yet, then I read this paper by Marty Chalfie about the idea of taking some DNA from a glowing jellyfish and splicing that in to any organism you want, to any protein you want, and hijack the cellular machinery of that organism to produce that protein with a fluorescent label on it. And you could have knocked me down with a feather. The bad news is that paper came out literally as I quit at Bell Labs. If I had seen that paper, I probably would have stayed in science at that time, because I couldn't believe how magical and transformative this was. And of course, those 3 guys got the Nobel Prize for it in 2008. But one of the biggest problems when I was trying to look at cells with near field was you just couldn't get the label on dense enough, you couldn't get it on specific enough, so even when the microscope was working, you just had all sorts of non-specific garbage on it and so forth. Here you have 100% specificity. It was amazing. And so I said, God damn it, I gotta do microscopy again. And so I tried to come up with an idea, because I figured I hadn't published a paper in 10 years. Who's going to let me in a lab? So I needed some intellectual capital. So I worked hard and used my physics and came up with this crazy idea, which turned out to be not so crazy in the end, about bringing a bunch of beams from different directions to coherently interfere to create a massively multifocal excitation field, to be sort of a confocal microscope on steroids to do 3D fast live imaging. So I started trying to get some place where I could build this. It wasn't going to take much, just a tabletop. And of course, the first person I went to was Harold, right? And I said, Harold, you want to go in with me on this? And he wasn't sure he was willing to quit his job to do something like this. And he said, he'd be chewing my cud, and he wants his own cud to chew. So, but he was willing to help me find a home. And so he took me to Florida State University, to the National High Magnetic Field Lab, which he had visited earlier. And there was a crazy guy there who was working with these fluorescent proteins, and might have a little lab space he could use for me to build my microscope. And we learned from him, Mike Davidson, that there was a new type of fluorescent protein that just came out. And instead of just being expressed and it's glowing, initially when it's expressed, it's dark. But if you shine violet light on it, you activate it, and then it's fluorescent. And so the light bulb, as we were sitting at Tallahassee Airport, popped on our heads, and we said, oh my God, remember that paper I published back in '95? if instead of doing it on the basis of different colors, if we turn down that violet activating light so low, then only a few photons will be absorbed by a few random molecules. Most likely, statistically, they'll be separated by more than the diffraction limit, and then we can localize them, turn those off, turn on another subset, and do that over and over and over again, and get a super resolution image. And so Harold said, okay, let's do that idea instead. Okay, and we said, yeah, okay, this one is trivial. Okay, so literally trivial. And that was what scared the hell out of us, because we're like, how come nobody's done this already? This is so obvious and so easy. And the good news is that, when I left Bell, I told them all to go to hell. But when Harold left Bell, he was able to take all of his equipment with him. So this was all in the storage shed, so we pulled that out. We put $50K each of our own money in it. And from the idea to having the functional microscope was 2 months. We were able to do it on the living room floor because he wasn't married, so there wasn't anybody to object to doing that. And then we had another problem, which is we have a microscope, but we don't know anything about biology. And so I had been scheduled to give a talk at NIH where I was going to try to pitch my optical lattice idea. And when I went there, I asked my host if I could please speak to Jennifer Lippincott-Schwartz and George Patterson, because they were the ones who invented this photoactivatable green fluorescent protein. And I swore them to secrecy at lunch and told them the idea and said, can we please bring our microscope here to try it out? And Jennifer said, sure, fantastic. I didn't realize at the time Jennifer says fantastic to absolutely everything, but we took that as a yes, and we brought the microscope, and within another month or two, we had images like this. This is a cut through a thin section through a couple lysosomes in the cell. This is the diffraction limited image. This is the photoactivated localization microscope or palm image. And to better appreciate the resolution gain, you go from this to this. And it's trivial. It's really trivial. It's just a little software and a wide field microscope, and that's it. And so, yeah, this took off like crazy, quickly. And that paper with the original idea paper was what got me to Stockholm. And so it also got Harold and I to Howard Hughes Medical Institute was building something that was going to be their own research campus. It was consciously modeled after Bell Labs. And so Harold and I were two of the very first people hired there. We went there and we started doing microscopy. So our paths diverged a bit. I stayed in the optics side. Harold, because of his experience in industry, had gone into the electron microscopy side, and we kind of went like this, and then around 2018 we joined again because of the problems that we were facing on each of our respective sides. So the first problem is, 99% of all the literature on super resolution microscopy is by looking at chemically fixed cells, because these are typically slow techniques. And it's been known since the beginning of electron microscopy that chemical fixation creates artifacts like you see there. You can see it in these EM pictures. This is if you do a high pressure freeze, which preserves ultrastructure, versus chemical fixation. The mitochondria get blown up, the chromatin in the nucleus is completely disrupted. But if you do it, again, high pressure freeze for super resolution, then you don't have this vesiculation here, and you can see the endoplasmic reticulum the way it's supposed to be. And so we said, okay, well, if we really want to do structural imaging with super resolution, we have to do with high pressure freeze. High pressure freeze, the ice is no longer vitreous if you go above liquid nitrogen temperatures. So now we're back in the cryo world. And then the other reason for doing this is because these two techniques, EM and super-res, are complementary. You get protein-specific contrast with the optics, and there's 20,000 different types of proteins in every cell, and it's those proteins that drive the function. But you want to understand, you don't have 20,000 different colors, so you want to have something to give you some global contrast to know where the fluorescence is in relation to everything else. EM staining gives you the global contrast. So you marry them together and you build a cryogenic, super resolution, fluorescent single molecule microscope. And when you get those images, you take that sample out, you embed it, and then you put it in Harold's microscope, which is takes a gallium beam to ablate off a few nanometers at a time of the block of the cell, and then do an EM image at 8 nanometer resolution that then ablate off another 8 nanometers, do it again and again and again, and get the 3D EM image. And then you can overlay them and get protein-specific contrast in the context of the global contrast of the thing. And so we had a paper in 2020 where we used this to look at various systems, first looking at endosomes and cells. And it's very difficult to follow the morphology of an endosome just by EM, but with the fluorescence help, we were able to show that they have just crazy morphologies as they start to bud off different compartments to go to other parts of the cell. We showed that between neurons, there's an interface with a protein JAMC that creates this sort of web-like structure you see there. And we showed that in the EM literature, it's generally believed that the dark regions in the nucleus represent transcriptionally inactive heterochromatin, and the light regions are transcriptionally active euchromatin. But there's also fluorescent markers of euchromatin and heterochromatin, and it turns out that they don't overlap in the same way. So that whole model of the EM, the dark means hetero and light means eu, is not true. And so we were able to show that. So all of that is fine, but I learned, I did some live cell POM around 2008, which was, finally reaching that goal I had back in '82 in graduate school to look at live cells beyond the diffraction. But it's a very limited tool because it takes a long time to photoactivate all those molecules. You're throwing a lot of intensity at the specimen, and cells don't like intense light. And so, yet I know at the same time that that you have to look at the dynamics. In fact, the only thing that matters, in my opinion, in a living system is the dynamics. Is that just like if I had the best high-resolution static images in the world, I'm still not going to understand the biology. It's less like I want to understand a football game if all I see is a picture of the quarterback getting ready to throw a pass, the halfback hitting a hole, and the cheerleaders making a pyramid on the sideline. I can't deduce the rules. But if I can watch the movie or go to the game, the rules become obvious. And so, you're never going to understand life by looking at it dead. You've got to look at it live. And so I heard that argument by Scott Fraser, who's now at USC, back in, I think it was like 2008, and I said, God damn it, I've been looking at dead stuff all my life. I'm done with that, okay? I'm going to be looking at living stuff. So you can do some live imaging with POM. It's called single particle tracking POM. And so what you do is, rather than trying to get a holistic picture of every molecule, you photoactivate subsets of molecules in a live cell, and you study their kinetics, and you watch how they diffuse or how they bind and so forth. And so this is an example looking at the raw data, and then this is two proteins. One is very mobile in the plasma membrane, so its tracks are very long. This one binds up into virus-like particles, so it gets very sticky. We used that a few years later to look at the Huntington protein in Huntington's disease, which has a glutamine repeat, which causes these aggregates that are known in Huntington's disease. The left side is the nucleus of a live cell without the disease. The right one is with the disease. And you can see that even the wild type has these little aggregates, but they're very tiny and very transient. And then the disease cell has the large aggregates you see, and we did labeling in a second color to do this single particle tracking of transcription factors, which are necessary to create proteins. And we found out that these transcription factors will get bound up in those aggregates like in quicksand. And so that means that they can't get to the DNA. That means they can't make the RNA. That means they're down-regulating expression of certain proteins. So it's certainly not indicative or conclusive in any way, but it does hint at a potential pathway for what is the significance of these aggregates in terms of down-regulating gene expression. So on the basis of those results and some other experiments we did with Bob Tejan, who was president of HHMI and now is my colleague at Berkeley, Tiege has been studying transcription through biochemistry through 30 years, and he understood the power of having these single molecule kinetics experiments. So in normal transcription, you have various proteins called transcription factors, which have to bind to the right part on the DNA, and then they recruit this polymerase, this big blue thing here, which spins along the DNA, soaks up nucleotides, and spits out RNA. So that's the process of transcription there, okay? So in order to initiate that, a whole bunch of proteins have to come together to one point in order to recruit that polymerase. So they had this model in their heads based on the biochemistry that it would take like 30 minutes for all of these things to come together and finally be able to start the process. But when we did single molecule imaging of the individual transcription factors, none of them were staying put anywhere for more than a couple seconds at a time. So it's like, well, how the hell does it work? And so the model that finally came about is that you have many, many copies of these transcription factors. At the start of the gene, they're kind of clustering all around like this, and they kind of come on like bees from a beehive, boom, boom, and sort of stochastically, and then that acts as a recruitment for the next one in the series and the next one in the series. So they're never all together like this. They're just kind of sequentially doing the dance needed to be able to recruit finally the polymerase and start the process. Well, it turns out that this process gets disrupted in many diseases because then the transcription factors are either too sticky or not sticky enough in order to then do the transcription. And so this is like, for example, a childhood cancer called Ewing sarcoma, which is usually fatal, is due to a translocation that takes an intrinsically disordered region of one protein and grafts it on to a different transcription factor protein and makes it extra sticky, which therefore means that it overdrives expression of the proteins that use that transcription factor, and that leads to this disease. So that then came up with the idea of, well, all of drug discovery is basically targeting about 2% of the proteome, ones that have this kind of lock and key mechanism. And so these intrinsically disordered regions present a different way of trying to modulate protein-protein interactions in the cell. And it's our belief that these are critically important and involved in many diseases. And so if we can instead look for chemical matter that modulates these intrinsically disordered regions and their kinetics, then this could be a new path to drug discovery to get to the other 98% of the proteome. And so we now have we started a drug company in Hayward, California, Icon Therapeutics, which is using incredibly fast, high-throughput screening of single-molecule kinetics. We can look at 600,000 compounds in several weeks on a particular target, and we're off to the races, finding hits and trying to find a new path to drug discovery. All right, so the other problem with super-resolution microscopy, and it's just natural, particularly for physicists who come into this field, to think about, I want resolution, I want resolution. Point of a microscope is resolution. No. Resolution is one thing you care about in a microscope. But if you're going to look at living systems, you care about whether you're harming the system. If you want to look at living systems, you care about being able to see the dynamics. And if the dynamics happen too fast, your resolution's screwed up anyway because all you see is a blur. You care about being able to not just look at single cells, you want to be able to look deep. But all of these things trade off against one another. If I want high spatial resolution, I need many pixels in my image. If I need many pixels, that means more measurements, it means more time, it means more light thrown at the spot. You get the idea. There's always these trade-offs. And so the history of super resolution is about a bunch of people marching like lemmings up to that point. And so again, once I heard Scott's talk about the importance of the dynamics, I said, damn it, I'm going to start marching in the other direction and see what I can find. And I was beaten to that punch by this guy, Mats Gustafsson, who was at UCSF as a postdoc and then finally a professor. And then I recruited him to Geneva in 2008. He had this incredibly beautiful technique called structured illumination microscopy, where you can get not 10x like palm, but you can get a 2x beyond the diffraction limit by illuminating the sample with grading patterns instead of uniformly illuminating it. And so if you've ever seen like a web chair or two screens against one another, you see those crazy fringe patterns. Those are called moire patterns. So those are low frequency patterns that actually encode high frequency information about the two screens that you saw. Or in this case, if you use structured light, they encode information about the sample beyond up to twice the diffraction limit. And so you can get beautiful images like this. Instead of requiring thousands of images, raw images to get one palm image, it takes 9 in 2D or 15 in 3D to get sim images. And so it's much faster, it throws much less, orders of magnitude less light at the specimen. And so it's a beautiful technique for being able to study dynamics at 100 nanometer resolution. Still not where you'd like to be at the single protein level, but it's a great complementary technique. So the bad news is after I recruited Mats, he was diagnosed with a glioblastoma in 2010 and he died in 2011. So I ended up inheriting his group and we kept trying to push the technology. One way is by using photoactivation to create additional harmonics to push the resolution down to about 60 nanometers, which we use to study clathrin-mediated endocytosis and the proteins involved in creating actin filaments inside of cells. And we've had probably about 50 groups come to our lab to do experiments with SIM. One is looking at myosin, which is a protein which binds together actin to create forces inside of cells. And so you have these little monomers that have a head and tail group, and they come together to make these bipolar filaments to draw in the actin. And there's actually 3 different isomers of these myosin monomers, and we were able to show, because it wasn't known whether they could intermingle in these bipolar filaments, and by using SIM, we were able to show that they could. We furthermore showed that while you can de novo create by just bringing in together monomers to create these big bipolar filaments, is it much more common, as you see here, whoop, that guy split off the first one. So if you have one, it acts as a template for the others, and so you get this avalanche type effect, which is really the way most of them are created. We were able to show in crawling endothelial cells that while it looks like the cytoskeleton is actually poking between cells, that actually what happens is in order to create a very tight structure, the leading cell leaves these little sticky fingers, which the trailing cell hitches to, and then models its cytoskeleton to exactly match the cytoskeleton of the other to have a very rigid structure. And then for the endoplasmic reticulum, which is one of the main organelles in the cell, which is involved in creating proteins from RNA, what you find is this sort of net-like structure, and then you also see these sheets at the diffraction limit. But it turns out that these sheets are not sheets at all. They're just tightly clustered little tubules that then collapse together and come apart. And with SIM, you have just enough spatial and temporal resolution to watch that process. So I'm going to skip this slide because I know I'm running over. I'll get to that point a little later. So all of that's fine, but now we have the next problem, is everything I've talked about is in a two-dimensional world. And we don't live in a two-dimensional world, and neither do cells. So it's a three-dimensional dynamic system, that every living thing is a pocket of reduced entropy, and matter and energy is flowing through it continuously. And so while structural imaging is important, the only way we're going to truly understand living systems is by studying them with high resolution in all 4 dimensions of space-time. And so how do we get there? Well, the good news is that the standard that people were using at the time for doing live imaging leaved a lot of room for improvement. There's 2 methods. The wide field microscope, which is basically what Hook and von Leeuwenhoek have been using for 400 years, where you just bring all of the light through the sample, and then the fluorescence is generated, and the fluorescence in the focal plane is in focus, the other stuff's out of focus, and you pick that up and put it to your eye or to a camera. In the confocal microscope, you use a laser to focus the light to a point, and then as the light comes back up, this out of focus light is discriminated against with a pinhole in another plane, so you only get the fluorescence from the spot in focus. You don't have all the out of focus light you have there. So in my opinion, one of the most important innovations of microscopy in the 21st century was when Ernst Stelzer, who was then at the EMBL, reintroduced a 100-year-old idea called plane illumination. So here you take a cylindrical lens to create a sheet of light which is coincident with the detection focal plane of the detection objective. And now, because you have that whole plane, there's no illumination above and below, so there's no out of focus light. But furthermore, by spreading the light across the whole plane, you can, rather than having to wait to scan this point along the plane, you can snap a picture instantly of the whole plane. And furthermore, because of having to focus that light in the confocal, in order to create an image fast, you have to have an intense spot. And so that creates a lot of destruction in the sample. So by having all the fluorophores radiating in parallel instead of serial, you greatly reduce the phototoxicity and photobleaching. So this has been transformative for understanding embryogenesis at single cell resolution. And so I was wondering, could the same principles be used to do subcellular resolution imaging of single cells? And there was an opportunity here because people were using conventional Gaussian beams to create images like this, where there's a trade-off between how long you can look through the embryo and how thick that sheet of light is. Well, I knew from my near-field days, there was a fad at one time with something called Bessel beams, where instead of illuminating with a uniform spot, you illuminate with a ring, and now you have two knobs, the diameter of the ring and the thickness of the ring, which can be used independently to adjust the length of the beam and the diameter of the beam. And so you can decouple it. And so now you can use that to create a narrower beam to look through cells. And eventually I realized that rather than using one beam, again, the best thing is to make multiple beams to spread them across to reduce the toxicity further. It turned out that the number of beams and their separation to optimally do this were completely predictable by that theory of optical lattice microscopy that I published back in 2005. So that's the thing that I abandoned in order to do the PALM. So I was able to pull all of that stuff out and then figure out exactly what we needed to do. And then we came up with the lattice light sheet microscope, which is not running for some reason. That's my life. Okay, hang on, let me see if we can. It's to go. There. Oh, no, he still doesn't like me. That's a shame. Okay. Shh. Hang on. Sometimes this thing is got to catch up, catch its breath. There we go. Okay. All right. So, so now we have a very thin sheet of light. We can scan through the cell, get a volume, and then do it again and again and again. And because the toxicity is orders of magnitude lower than a confocal microscope, and because it's so much faster because we're taking an image at once, and because industry created incredibly fast cameras, we're able to now image cells for thousands of time points, hundreds of thousands, if not millions of single 2D images to be able to look at dynamics of cells. So, you know, I am morally convinced, I'm very proud to have won the Nobel Prize for PALM, but I'm morally convinced that this will be the most impactful microscope that I ever make. Because we've had over 100 different groups come to my Geneva lab to use this. People who've been looking at the same system for 30 years and they see it in a completely new way. You know, I've never felt more like Galileo, you know, with his little telescope seeing, you know, moons of Jupiter and stuff like that than I have with this microscope. Everybody sees things in a way. And they all go away with 10 terabytes of data, and then a month later they end up calling us crying because they have no idea what to do with 10 terabytes of data because they're bad. And that is the Achilles heel, and it's still the Achilles heel, except the questions now are not 10 terabytes anymore, it's 10 petabytes. And that's what the big problem is. So I'm going to skip this one. Lots of applications, and my computer is not happy. Come on, man, you can do it. Okay, anyway. Oh, no, it just crashed altogether. So that's good because then I can get the cobwebs out and we can start again. Oh, I'm already at 12, so I better go fast. Hang on. Which one do I have right now? I don't even remember. I'm a space bug, so that's probably what's on that screen. Now I don't even have a mouse. Well, that's unfortunate. Hello, mouse, where are you? Let's see. Okay. Come on, man, you can come back. I'm sorry, folks. Got it? Good. Okay, except I'm way at the beginning. Okay. All right, so anyway, so I will talk as I scroll. So with the lattice light sheet microscope, like I say, we've had 100 different groups come to use that microscope, and the data is the problem. But then there's another problem, which is this, okay? We started applying it not just to single cells, but to trying to study subcellular dynamics in multicellular organisms. And the reason for that is a cultured cell on a cover slip is a completely unnatural object, right? I mean, cells didn't evolve on cover slips, they evolved in organisms. And so we know from biology that the phenotypes we observe are the result of gene expression. And gene expression is influenced by environment. So if you have the wrong environment, it doesn't matter how good your microscope is, how can you trust the phenotypes you observe? So you have to take the cell away from the cover slip. The problem is that multicellular specimens have different refractive indices for nuclei, cytoplasm, and everything else. So trying to image in them is like trying to image through a bag of marbles. Not exactly easy, or like water on your windshield. The good news is, I always say, I give a lot of talks about the historical connections between astronomy and microscopy, and the take-home message of that is that microscopists are the retarded stepchildren of astronomers because they invent everything we use 50 years before we use it. So another one is two ideas here. First one is that this is from the Hale telescope, which was, when I was a kid, the biggest telescope on Earth of the Crab Nebula. Well, that's fine, but it's nowhere near to what the real resolution that telescope should be able to create, because the atmosphere ends up scrambling the light to a degree and create aberrations that limit the resolution. So they said, well, we'll put our telescope into space. So that's the Hubble telescope from, went up in '86, and then, this is a picture from '90, but, and then with that, this is a smaller mirror than this mirror by a factor of 2, but you can see how much better the resolution is once you got rid of the aberration. Well, there's an analogy to that in microscopy, and it's called clearing. So you take a dead tissue and you bring in chemical treatments which homogenize the refractive index so that you don't have the aberrations in the image like that. One of the most interesting types of clearing is something that Ed Boyden's group at MIT developed called expansion microscopy. So there you not only clear the specimen, but also it's in a polymer which has these chains to it, and then those chains are initially compressed, and then you change the osmotic balance of the solution it's in, and then the sample expands. So now your diffraction-limited microscope is now a super-resolution microscope because your sample itself expanded. And this is the same fly brain, it's just 4 times expanded. It's the same microscope resolution, just blown up. And so then we can use that then to do look over very large fields of view at super resolution that you would never be able to do with Palm, with the caveats that there are potential artifacts created by this expansion process. And so we've been able to use that to, it's acting up, whatever. I would go across that. But anyway, it's a good complement. At Genelia, they're trying to do the connectome of an entire fly brain, 100,000 neurons, to try to understand how that works. It's a very, very difficult problem. With expansion microscopy, you can do that in a way that you won't be able to see all the neurons, but you can learn a lot by looking at subsets of neurons and certain subcircuits, and that's all of that. And so but the problem is I think I'm on firm ground when I say that nobody's going to be doing expansion microscopy live anytime soon, because none of our bodies would want to be expanded 4x no matter what you do. Okay, so I mean, I may get there eventually, but through natural means. So then you come up with the other wrinkle astronomers have, which is adaptive optics. So what they do is they shine a laser into the stratosphere to excite sodium atoms to create an artificial star, the light of which comes back into their telescope. And that star gets warped by the aberrations of the atmosphere. They put that into a detector to measure what that warping is. And then they change, that's this guy, and then they change the shape of a mirror that the light bounces off in as it gets into the telescope to exactly cancel the warping due to the atmosphere in real time, and then get back to the diffraction limit. So with that, you know, now like the Keck telescope has actually better resolution than the Hubble Space Telescope in the infrared because it has a bigger mirror. In both cases, we're down to the mirror, but now we can do this with a ground-based telescope instead of a space telescope. They're running out again. Okay, here we go. I'm sorry, guys, but I don't know what's happened to my computer today, but it's not happy. Anyway, we've applied that a lot, so I'll just say a couple things about what happened with okay, we're not going to have pictures, but I'll tell you what the end of that is. So we applied adaptive optics first in confocal and two-photon microscopy. We're able to show that it was effective. And then we said, okay, well, we got to do this with the lattice light sheet because that's much faster, non-invasive, so forth. And so in that case, you actually have to have 3 microscopes in one because you have your lattice light sheet, you have an adaptive optic loop to correct the detected light, the fluorescence light that's coming there, but you also need an adaptive optic loop to correct for the light that's going in. And so you have a very, very big microscope that covers a 10-foot optical table and costs well over $1 million. And so when we built the lattice light sheet, a big part of my group has been about trying to get other people, biologists, on board. I mean, one microscope in one lab is not doing anybody any good. It's too narrow a bottleneck. It has to be spread out. And so the ultimate end of that has to be commercialization, because only commercial vendors will have the support, the service, everything, being able to make everything user-friendly enough that biologists have a turnkey instrument they can use. But in order for them to do that, it's a $10 million investment, and so you have to find out, you have to prove to them that there's a market for that investment. And so that's what bringing the 100 groups in with the lattice light sheet was all about. And part of that process was also to create a fully documented version of the microscope so that the other groups could do that. So currently there's about 150 different groups who have licensed the plans. I would guess there's probably about half that many have built lattice light sheet microscopes, and that was enough to get Zeiss to license the lattice light sheet tech, and now there's a commercial version of that. So it's kind of off to the races, still constrained by the data. We wanted to do the same thing with the adaptive optical lattice light sheet, because nobody's going to want a 10-foot train rack that costs $1 million. So we redesigned that microscope from scratch to be smaller, cheaper, easier to build. And when we did it, we said, well, to do AO, you need this hardware. To do lattice light sheet, you need to do this hardware. And you say, hey, wait a minute. I use that same hardware to do confocal. I use that hardware to do two-photon. And you realize that you have already paid for all the hardware necessary to do pretty much every microscope that anybody has designed since 1980, okay? And you say, well, why not make it a Swiss Army knife then, okay? You know, try to get the most utility out of that money that you've just spent. So we've made this microscope called Mosaic, which Gokul is one of the prime guys on, to basically make this Swiss Army knife box for around $400,000 that can do any one of these techniques and do each one of them with adaptive optics so they can be extended to multicellular systems. And so that's kind of where we're at right now. and about 40 groups so far have signed research licenses to build their own. I'd say probably about 15 are well into the build, if not done. We've just basically finished everything on it and trying to write a gargantuan paper about its utility right now. But yeah, the mosaic is the big thing, but the biggest thing is that the mosaic is even more productive in data taking. So we can easily get 100 terabytes a day of data. And this doesn't make sense. It's just, there's just no way to really be able to analyze and handle data at that scale. And that's become the biggest bottleneck problem. Gokul is just the right guy to try to figure out that problem. We have our own very powerful cluster and we have great computational scientists, but if anything is going to kill this kind of thing, that's going to be it. But in the same time, you know, at lower scales and so forth, I'm convinced that these tools are going to create a lot. So anyway, I'm not even going to try to get that damn thing to run again, but I'm sorry about that. So you didn't get to see those final slides, but I'm convinced that, you know, while the 20th century was really the story of biochemistry and structural biology and molecular biology, I really think that with effort, that these imaging tools are going to really launch a new growth in our understanding of biological systems by being able to understand the findings of those three fields in the context of dynamic living systems. Okay, thank you very much.
Thank you very much, Alex. So that was amazing, the whole journey. And just to make it fair for the people who are behind the computer screen, so we're going to probably, do you have any questions online? Not yet. Okay. So anytime we have interpreted to us, we'll have people from the audience to ask questions, and then when we will have somebody will interrupt. Okay. Questions?
Yeah. Oops, sorry.
What does it take to build this microscope? I can think of a lot of uses in my own research that I could use it, but
Yeah.
there's probably a technical ability and a
Yeah.
pricing
It
component.
depends on the microscope, right? I mean, to build a palm microscope, it doesn't take a hell of a lot. To build a lattice light sheet, you know, that you have to have somebody who's a good engineer, physicist type of person to be able to assemble and maintain it. And in fact, you know, I was just arguing this morning that that's another problem, right, is incentives. Because often I've seen now with 150 licenses what happens, right, is that there's a biologist who's seen what it can do, wants one for themselves, they have the money, they hire some physicist engineer type to build it, the physicist engineer type builds it, kind of is bumping up against a career limit there, right? I mean, if it's for his degree, he wants to do something novel of his own to be able to move on. If he's doing it otherwise, he just kind of feels like he's at a dead end. But so pretty much after 5-6 years, this person is somewhere else, right? And then if you don't find somebody to take over, the microscope just languishes. And I've seen that happen over and over again. Yeah. The what? It's not complete. There's no such thing as ever getting, and until we don't throw any photons in it, there's always a risk, right? So, but we've come orders of magnitude from where the field was in the 80s. That's for sure. And, you know, by almost any metric we can find, you know, redox or, you know, how many rounds of mitosis you can go through, or, you know, dynamics of things that, you know, like microtubule growth rates and like that, that we're in the physiological realm. And if we go back to anything even close to the old methods, you see it's not pretty. Yeah.
Yeah, this is Jai from Bioengineering. That's a fantastic talk. I learned all in my class within 1 hour now from history. So I highly appreciate your recognition of dynamics on single molecules. But the field in general, the single molecule field, people have different language with population molecule language. So for example, protein interactions, people use binding in the population level, and the single molecule level timing.
You have ensemble type of molecular, and you have singles level.
So in general, do you have any feelings as how many molecules, single molecules, we need to observe in order to
Get statistical significance? Yeah. It really depends on the question, right? So that's a big question we ask ourselves at our drug company, right? Is exactly what kind of statistics we need in order to have statistical significance of, that your compound is modulating, right? And so it's a problem-specific question in that you have to know exactly something about the system to know exactly what kind of statistical significance you need. Okay, that's all I can say. Yeah.
Okay, let's partially answer my second question. The drug screening, what's the cutoff in single molecule screening? In the population, we use some kind of reductions, Ki and so on.
Yes.
Population.
Yes.
But single molecule level, what's the cutoff?
Basically, all we're really looking for is to look at what are the outliers of the kinetics and what's the statistical significance of those outliers? Because those are our leads, right? And then we start working with those, and then we do standard, biochemical assays on those particular hits. So we do an initial screen through the single molecule, then we go to that, and then once we have something which from the biochemical assays looks significant, then we go back to the single molecules in order to do structure-activity relationships, right? Because then we can modify the compound a little bit and find out exactly how they connect. And that really speeds up SAR, structure-activity relationship definition, because you can very quickly work through any perturbations you do to the compound and see how it affects the kinetics.
Thank you, Dr. Betzig, for a wonderful presentation. I particularly like the part about you quit your job twice and still became a Nobel laureate.
That's right.
I'm not so sure that's the formula, but But I'm really excited about the data analysis, big data problem. It looks like you have. Can you elaborate on what you intend to do in order to analyze this data sets?
Well, again, the best way is what we've done so far, right? So Wallach and Gokul played a huge role in this, right? Is to write scripts that can, even just forgetting the analysis, the processing of the data takes quite a while, all right? I mean, do the deconvolution, the stitching, and all of the rest. We now have that where we can do that in real time, even at these rates of where we're getting, 50 terabytes a day. But for the analysis part, that's the other big problem, is that every question a biologist asks is different and requires some kind of different type of code in order to get the answer. If it's particle tracking, then it's easy. I mean, again, through ICON and through elsewhere, there are very rapid and real-time particle tracking things that one can use. But when you get into, for example, trying to understand the morphology of mitochondria and like that, you have to figure out some kind of scoring. You have to, develop the code in order to do that scoring. And you have to do it in a scalable way that can go through the amount of data you have. And every one is just a custom job. And the other part of it is it really requires a tight interface between two communities that don't talk to each other well, which is the biological guy who has the question and the computer scientist who has to figure out how to take the images and reduce it to what the biologist finds as meaningful. And that's why we developed the Advanced Imaging Center at Janelia, and now the Advanced Bioimaging Center at Berkeley, was to directly address those questions. And it has been a struggle. I mean, it really is. It's like, it's a multi-year struggle for almost every question that's asked. We have papers coming out now from collaborators for images we took from them in 2012. It's really at that level. Okay, so I wish I had an answer, but I don't. It's just the diversity of the biology alone on top of the complexity and size of the data is just really daunting. And frankly, I'm quite discouraged right now. I mean, I was all gung-ho a few years ago we'd get there, and now I'm just not sure that I'm up against another one of those things that's gonna get me to quit a third time. Yeah.
Other questions? Do you have anybody online? Okay, so before we finish, I want to ask you to even in a broader scale. So we focus on living systems, but there are a lot of interesting dynamics that is in non-living systems and interface living, non-living. I'm talking artificial photosynthesis and all this biomimetics, and looking at transients, because sometimes you look spectral resolution, sometimes spatial resolution, but how do you see expanding in that direction?
No, not at all. I'm totally enamored to, I've done a trajectory, right, through my career of going from first, looking at truly dead things like semiconductors and magnetic materials, to looking at single molecules which are not alive, to looking at cells which are alive but artificial, to looking at whole organisms, but whole organisms which have been, they're model organisms that have been adapted to be able to look at in our instruments. And they're not the way natural evolution created them, okay? If I were to continue in microscopy, and if I were not daunted by these data problems, I think the next frontier is really to push advanced microscopy intravitally, okay? Yes, and other mammals, at least, right? I was really interested in that idea about 6 months ago, but I'm tied to Berkeley, and Berkeley is not the right place to do that because we just don't have you'd have to go across the bay to UCSF for biopsies, and for vivarium and like that, we're very limited. So, yeah, I'm actually starting to put my toe in the water of really jumping off that cliff for a third time. Somebody asked me what my background is, and Linda, that is the Starship. Okay, you're seeing the upper stage and the booster being mated together in that picture there, right? So I'm a space buff and I'm a nuclear power buff. And in fact, I think that if we don't get on the stick about being serious about nuclear energy as a component of our future, solar and wind is just not going to be enough. And we're looking for many, many years. And particularly if we want all the people in the poor parts of the world, in Africa, to have our standard of living. Yeah, I mean, Elon Musk just gave his talk a couple days ago about his vision for a complete, and I love Elon Musk. Nobody loves him more than I do. But I think solar and wind is a complete solution to our problems by 2050. It's fantasy. And in fact, if you believe that there's significant risks of climate change, I'm here to tell you now, without significant adoption of nuclear, they are not going to be solved in this century. And the die will be cast, and whatever happens, happens. So there's a part of me that says, I can continue to work on microscopes, and I'm good at it. But that's not where the biggest problems lie. And if anybody has, I guess, capital to take a risk and try to push in an area that's truly important, that maybe I should use that capital and try to push in that direction.
Other questions? Okay, let's thank Eric again for